Hello, my name is Bunik and I'm from Fluidam. Welcome to my poster on a streamlined immune profiling workflow using an automated acquisition system and the MaxPar direct immune profiling system. As a quick introduction to our technology is Mass Cytometry and we have a couple of versions of the instrument. Recently, we have launched the new set of XT, which is our fourth generation of Mass Cytometer. The older generation is called the Helios system. The site of mass cytometry allows for highly multiplex single-cell analysis of more than 50 plus different protein markers simultaneously by using isotopically pure metal-labeled antibodies to stain cells and gain different marker expression information. A significant advantage of the CyTOF is the ability to resolve more than 50 markers in a single panel without the need for compensation, making mass cytometry an ideal solution for routine enumeration and phenotyping of immune cells. The MaxPar Direct Immune Profiling Assay, or MDIPA in short, together with the MaxPar PathSetter software, were developed as a sample to answer solution for human immune profiling using mass cytometry. The MDIPA kit includes an optimized 30 marker panel contained in a lyophilized single tube format. The MaxPar PathSetter is an automated software to report population statistics, staining assessment, and relevant data plot. With the 30 marker panel shown here and coupled with the MaxPar PathSetter software, we could identify 37 different immune population from your either whole blood or human PBMC samples automatically in just 5 minutes with a press of button. These 37 different immune population includes all major subset of your T cells, 2 gamma delta T cells, as well as mid cells, NKT T cells, together with other population of monocytes, granulocytes, dendritic cells, which also includes your rare plasmacytoid dendritic cells. Also keep in mind, this 30 marker core panel still have 14 open channels if you would like to add more custom markers to the core panel for more subpopulation identification. The MDIPA was originally validated on the Helios Mass Cytometer. So in this poster, we will do a comparison data with our newly launched CyTOF XT and compare it with Helios. So jumping into the materials and method, I would like to spend some time to first talk about the new mass cytometer, the CyTOF XT. We have recently launched this instrument at end of March during our Mass Cytometry Summit. This new generation of mass cytometer have a novel design and is equipped with a fully automated sample acquisition system. We also did a major revamp of many of parts of the instrument to make it easier for operator to operate the instrument. For this part, I would like to focus on the automated sample acquisition part. Here we have a new auto sampler which consists of 4 new major components. The sample probe, a syringe-based pump unit which is inside here, a bottle station for acquisition and cleaning solutions, and a carousel inside here which is able to hold 13 sample tubes in a chilled 4 to 8 degrees Celsius inside here. The auto sampler modules automate the following process. First, tuning of the instrument, cleaning of the sample fluidics, acquisition of samples that is already in suspension. It can also do resuspension, addition of EQ calibration bits, and acquisition of pelleted samples, as well as automated washing after one sample acquisition before proceeding to the next one. It also has an automated clot detection and automated removal of clots. All these functions essentially combined together would mean that you as a user just have to load samples, set up the samples inside the instrument, and then press start. And you can then walk away from instrument and do other things while the instrument does the sample acquisition in an automated manner without requiring any hands-on from your side. For experimental setup and comparison between CytoXT and Helios, we collected one whole blood samples from one healthy donor and also acquired two healthy human PBMC from stem cell technologies. So this for the PBMCs were taught using CTL anti-aggregate watch 20x solutions from cellular technologies. We all do this according to the manufacturer's instruction. For each donor, 12 MaxPar direct immune profiling assay tube were used for staining. Staining and acquisition proceeded as outlined in our MD IPA user guide, with a following exception. All samples were washed using the MaxPar Cell Acquisition Solution Plus for CytoXT. After the first Cell Acquisition Solution Plus wash, replicates of the samples were pulled and redistributed in order to control for tube-to-tube variability. Triplicates of the samples were acquired in parallel on two Helios instruments running on the side of software version 7, and also the other two were acquired on the side of XT instrument running on the side of software version 8. Normalized files were then analyzed using the MaxPar PathSetter software. Now, jumping into the results, a quick eyeball look at figure 6 shows that CytoXT and Helios produce comparable and repeatable results. So you can see in yellow represent data from the CytoXT, while in purple represent data from Helios instrument. Looking at the bar chart here, we see comparable population frequencies was detected between different donor and on different instruments. This also include the rare population like plasmacytoid dendritic cells as shown here. Now looking at table 1 here, for a bit more details, you can see that there are percentage population frequencies detected between these two different instruments are comparable. So if we look at all the major T cells and population here, you can see comparable mean and also percentage series obtained from different donors here. So if we look at more smaller rare populations like your plasmacytoid dendritic cells, also comparable mean and percentage series were obtained between different instruments and different donors. With the results, we also did a Sensei Plot, which is a clustering analysis and also a dimensional reduction tool. This is performed also inside the MaxPar PathSetter software. How you look at it is that each islands represent a population of cells. And you can clearly see that these similar results were obtained between the CytoXT here versus the Helios instrument here, as all show comparable population islands here. In this analysis, do take note that we also intentionally exclude the neutrophils from the Sensei plot for better visualization of other cell types, as neutrophils are present in very high amount. So having neutrophils in this plot would cover all the smaller population frequencies here. Next, we did a dimming regression analysis on the mean population frequencies from whole blood and PBMCs samples from CytoXT and plotted against Helios to show that CytoXT and Helios generate equivalent population frequencies. The H0 test shows that the slope equals 1 was not rejected, indicating that there was no statistical difference between the population frequency analyzed from the files acquired using these two different instruments. If you look at this picture here, the shaded area in red indicates the associated confidence limit bound. The 95% confidence limit of the slope are shown for the line of best fit here. While there were no difference in population frequency while there were no difference in population frequency detected between both sides of XT and Helios, we found that our new site of XT instrument, this actually improved staining assessment value, indicating that site of XT actually generate improved staining resolution. Using our automated software analysis for the MD-IPA panel, which is the Maxma Pathsetter software, we perform a staining assessment based on statistical approach called strictly standardized mean difference. This is represented by a beta value. How to read this beta value is that a higher beta value would mean greater resolution between the positive and negative population. This is calculated using this formula here, where MAD equals mean absolute deviation, POS means positive population, and neg equals negative population. On figure 9b, here we perform a plot of the average beta value from site of XT acquired flowers and plotted against the beta value from Helios acquired flowers. This was plotted using the values shown using the values here on table 2 here. We did a dimming regression analysis and performed to compare the staining assessment between these two different instruments. The H0 test that slope equals 1 was rejected, and the upper and lower 95% confidence limits are more than 1. This indicates that site of XT on average will have a higher beta value as compared with the Helios. The shaded area, which is red, indicates the associated confidence limit bound. The 95% confidence limits of the slope are shown here for the line of best fit. So as a conclusion, this site of XT is a new generation of site of instrument that shares the same reliable level of performance as Helios when using the Maxma direct immune profiling assay. The site of XT and Helios acquired files analyzed in the Maxma Pathsetter software resulted in no statistical significance between these two platforms. The site of XT overall resulted in improved staining resolution for whole blood and PBMC samples as compared with the Helios. Ultimately, the hands-free acquisition on-site of XT and the automated analysis of the MD-IPA panel enables you researchers for a more streamlined high-parameter immunophenotyping of whole blood and human PBMC samples. With this, I thank you for your attention and happy to take any questions. Thank you so much.